So we'll be discussing this paper in this video and introduction and background. So context and importance of the study. Fear responses to predator odors are innate and crucial for survival. Rats as prey animals exhibit strong defensive reactions when exposed to predator odors. Example, cat, urine, or fur. Previous research shows predator odors trigger behavioral, endocrine, and cardiovascular changes in rodents. The study aims to measure both behavioral, freezings or avoidance, and cardiovascular, heart rate, blood pressure responses to cat odor. Okay, so previous research and gaps. Past studies focus primarily on the behavioral responses to predator odor. Limited research has been done on cardiovascular changes in rats exposed to predator scent. This study integrates both behavioral and cardiovascular responses to provide a holistic view. Right. So hypothesis and objectives. So key hypothesis: rats exposed to cat odor will show significant fear responses demonstrated through freezing behavior and cardiovascular changes like an increase in heart rate or blood pressure. Right. Now, objectives of this study are, there are three main objectives. Firstly, to examine behavioral responses, freezing, avoidance in rats exposed to cat odor, to assess cardiovascular responses, changes in heart rate and blood pressure, and to determine the duration of these responses, whether they persist after removal of the odor. Right. Now, methodology, subjects and setup. Subjects were used, the male Sprague Dolly rats were used, specific breed chosen for uniformity. Setup, rats were placed in an open field chamber equipped with sensors for movement tracking and cardiovascular monitoring. And there was also a hole or a opening in which the rats could go in order to hide. Right. Now, experimental conditions. Three groups were studied: cat odor exposure, clean collar control, and no collar control. So, cat odor exposure, rats exposed to collar worn by a domestic cat for a prolonged period. Clean collar control, rats exposed to a clean odorless collar, controls for novelty effect. And no collar control, rats exposed to no stimulus, baseline control. Measurement techniques. Behavioral measurements. Freezing behavior, defined as a complete lack of movement except for breathing. Avoidance, measurement measured by time spent in the opposite zone of the chamber, away from odor, where we have the hiding spot or the hole. Right. And cardiovascular measurements, heart rate and blood pressure, HR and BP, recorded by telemetry implants. So results and data analysis. Firstly, behavioral responses. So we have the freezing behavior of the rat. Rats exposed to cat odor showed significantly higher freezing duration than both control groups. Graph 1 shows the freezing duration versus condition graph. Cat odor group, long freezing periods, 50 to 70% of observation time. Clean collar group, minimal freezing, 10 to 20%, and no collar control, almost no freezing, 5 to 10%. Now, avoidance behavior. Rats in the cat odor group spend significantly more time in the opposite zone of the chamber because they, since they smell the odor of, let us say, the cat, which is a predator. So a fear is induced in their mind that a cat may be nearby, right? So that's why they would go in the opposite zone of the chamber where the hole is placed, where they can hide, right? Now, graph 2, time spent in opposite zone. Cat odor group 70 to 80%, clean collar group slight avoidance 40 to 50%, and no collar control, even distribution around 50%. Now, cardiovascular responses, heart rate change, significant HR increase in rats exposed to cat odor. Baseline HR, baseline heart rate 350 beats per minute before exposure, sharp increase to 450 beats per minute immediately after odor exposure. So heart rate gradually decreased but remained elevated for at least 10 minutes. Now, blood pressure changes. Blood pressure spiked following odor exposure. Graph 4, blood pressure over time. Baseline BP 110 millimeter mercury Hg increased to 140 mmHg immediately post exposure. Decline was stayed above baseline for 10 to 15 minutes. Now, discussion and interpretation. So, key findings were: firstly, strong behavioral response. Freezing and avoidance confirmed that cat odor induces fear in rats. Second, elevated cardiovascular activity, increased heart rate and blood pressure indicate autonomic nervous system activation. Sustained effects, both behavioral and cardiovascular responses persisted beyond initial exposure. Now, implications. It confirms predator odor triggers both psychological and physiological fear responses in prey animals like rats, in this case, and cat is the predator. Suggests predator odors can serve as strong natural stressors in research on fear and anxiety. Findings may apply to human PTSD research since rodents are used as models here. Right. Now, study limitations. Only male rats were tested. Females may respond differently. Single exposure session. Long-term effects unknown. Specific predator odor used. Other predator scents like fox odor were not used. So it might trigger varied responses. In which, like for example, fox odor has TMT. So that's a different odor and it will show varied responses. Conclusion: rats exhibited strong fear responses to cat odor, both behaviorally, freezing avoidance, and physiologically, heart rate, blood pressure increases. Study bridges gaps between psychological and physiological responses to predator odor. Future research should explore long-term effects, gender differences, and multiple predator odors. And that's it for this video. Thank you guys for watching.